Hello everyone, welcome to Visionary Classes. So today we'll discuss on the Unit 2 and today's Unit 2 topic will be pH. Okay, so Unit 2 from UGC NET. UGC NET Environmental Science. We are discussing on the topic of environmental chemistry, that is pH. So basically what is pH? pH is regarded as power of hydrogen. Okay, and what we have seen till now in the pH in our primary classes, that pH scale is basically divided into two types, that is strong acid and strong base. Means we have one acid category and one base category. So whenever the pH is around 0 to 5 or 6, it is considered as acid part, while it is 8 to 14, it is called as base part. So basically the 7 is considered to be neutral. Okay, so you can see this is the pH scale we have seen till now. So what we will discuss here, the first thing we will discuss about the title, what it describes. Now then we have seen the content. This is the content part. Overview part is also called as the content part. This is the content that we are discussing in this part, and this is the page number 2 here. Okay, then we will discuss about the pH introduction, means what is pH, how it is called as pH, that we will discuss in the pH introduction. Then we will go to the pH scale, how the scales are divided into different parts. Then finally we will discuss about the formulas related to the pH, how it is determined, and the measurement techniques that will be used in the pH measurements, that we will discuss in the next part. Okay, so let us discuss the first one. So what is pH? Exactly what is pH is very interesting to know. Okay, so pH is what? So pH is basically is a measure of the concentration of hydrogen ions H plus in a solution. This solution could be anything, water, okay, and some liquids. Okay, so water, some liquids, whatever it may be, pH is basically the measure of concentration of hydrogen ions present in that solutions actually. Okay, indicating its acidity or alkalinity properties. Okay, so the term was coined by a Danish chemist Sorensen in 1909. Okay, it comes from the German Potenz von Wasserstoff, meaning power of hydrogen. This is the phrase that says that it is a power of hydrogen that is regarded as pH. So it comes from a German letter, German phrase. Okay, so it is basically a German phrase where P denotes the power or exponents. Exponent, okay, exponential power. That's why we use exponential forms of this pH in our formula. So here you can see there are different types of materials that we considered in different pH. So basically pH 0 is the most acidic one, then comes 1 and 2. In 1 we have gastric juices, gastric juice like our in our body itself, these juices are present, that is gastric juice. Then in 2 it is lemon juice, in 3 to 4 it is wine, while 5 it is coffee, 6 to 7 everyone, that is milk, pure water, blood, these are above 7 and between 8. Okay, sea water also. So basically these are the typical pH that we are seeing for different materials. Then comes from 10 to 11 we have detergent. Okay, 12 it is household ammonia and bleach. Correct. So acidic solutions have pH value less than 7. That we have a conclusion that acidic solutions always show a pH less than 7. That we are can see. Then basic solutions have a pH values greater than 7. Here less than 7, there greater than 7. This is the typical thing that everybody knows from the beginning. But the thing that we are getting out from here is that it is called as power of hydrogen, and why it is called as power of hydrogen, and from which letter it has been coined, and from which phrase it has been derived that it is a power of a hydrogen, and what are the basic types of liquids that are showing different pH at different scale. Okay, these are the things that we are getting out from this part or this slide of the presentation. Okay, so now if we consider the formula, the typical formula that we use in pH is minus log H plus ion, where H plus ion is also called as 10 to the power minus pH. So what is log? Basically some of the students don't know what is log. Okay, so a logarithmic scale, if we determine, if we say log A equal log AB, where A is the base, log AB, if we consider like this, equal to C. Okay, if we consider like this and we say it is a C, then we have C, then we have A to the power C equal to B. This is the thing. It is simply like this. Okay, so it implies same thing as it is written here. Okay, so here basically in the log we have base 10. That's why we are writing it as 10 to the power minus pH is equal to H plus ion. So whenever there is a question and it is given that you have to find H plus ion, if pH is given, okay, if pH is given and you have to find H plus ion, then you will use this formula. And if you need POH ion, then you will use this formula. Okay, this is the simple thing that you should know before you get into the answer. You should know this is the simple formula that we consider for our calculation of pH and POH. Okay, so here we can define, now you can say that what is pH and what is POH in a formula. Okay, so if we consider, there is one more formula that I have not written here, but I will show you. There is one more formula that you should know before we will go out of this slide, that is pH plus POH equal to 14. This is the formula. It is at what temperature? It is at 25 degrees Celsius. Okay, so this is at 25 degree Celsius. So from here, why it is written as 25 degrees Celsius? Because pH is a temperature dependent. Okay, so pH is a temperature dependent. So what happens? So there is a relation that whenever pH is high, it means temperature should be low. Temp should be low if pH should be high. So pH is basically inversely proportional to the temperature. Correct? pH is inversely proportional to the temperature. Means acidic conditions gets more easily in cold temperature compared to hot temperature. Okay, this you can learn. That I should not discuss much more about these things, but you should know that pH is basically dependent on our temperature, and how it is dependent, it is inversely proportional to each other. Whenever pH goes up, the temperature decreases. So whenever temperature rises, pH decreases. Simple. There is no much concept behind this. Okay. So now we'll discuss about the pH scale. So here you can see we have already discussed those things, but again we are seeing that 0 to 1, this is HCl, this is stomach acid, this is lemon, this is vinegar, this is wine, this is coffee, this is milk, this is water, this is blood, this is baking soda, milk of magnesia, ammonia, soap, bleach, drain cleaner, etc. So here we have seen a lot of things that we are using in our daily life. So this is basically the pH scale of different materials. Correct? So here you can see the pH scale is divided into different parts. So the first part we are seeing here, that is pH 0 to 1. pH 0 to 1 is a strong acidic, having example hydrochloric acid and stomach, and pH 2 to 3 is acidic, vinegar and lemon juice. pH 4 to 5 is weakly acidic, like tomato juice and beer. Sometimes rainwater also consider here. Rainwater also comes in between this part. So pH 6 to 7 is neutral to slightly acidic, like pure water and saliva. pH 7 is exactly neutral, pure water. pH 8 to 9 is weakly basic, baking soda solution and seawater. pH 10 to 12 is basic, example soap solution, bleach. pH 13 to 14 is strongly basic, example sodium hydroxide solution and drain killer. Then comes each whole number change in pH represents a tenfold change in the hydrogen ion concentration. That is simple, because we have seen that log H plus ion concentration is equal to pH. Okay, and then we have also seen that H plus ion equal to 10 to the power minus pH. So obviously it will be tenfold times. In the formula itself we have seen that. So a lower pH indicates higher acidity, while a higher pH indicates higher basicity. Correct? It is good. The midpoint of the scale pH 7 is neutral and correspond to the pure water at a standard temperature and pressure condition. Then again it is told that it is pH 7 at a standard temperature. So that is 25 degrees Celsius only. Okay. So let us see the next thing. So there is a formula related to pH, that is Henderson-Hasselbalch equation. So what does it tell? That pH is equal to pKa log A minus upon HA. Okay. So what is HA minus? A minus is a conjugate base, because it contains no H plus ion, while this is conjugate acid because it contains H plus ion. Okay. So the relationship between pK and pH is crucial in understanding the behavior of acid and bases, particularly in an aqueous solution. This is the fundamental thing that you should know. So it is telling that whenever pH is equal to pK, the concentration of acid and its conjugate base are equal. This is known as half equivalence point. At this point, the solution will have a buffer capacity, meaning it can resist a changes in pH. And whenever pH is less than pKa, the solution will be more acidic than pKa value, indicating that acid is predominantly in its undissociated form. Okay. And whenever pH is greater than pKa value, the solution will be more basic than pKa value, indicating that acid is predominantly dissociated into its conjugate base. Okay, so this is the concept that you should know. Means whenever there will be solution will be more acidic, whenever the solution will be more basic, it all depends on its pKa value. Okay, it all depends on pKa value of our acid and base. Okay, so this formula that you should know before you are attempting something else. Okay, so then comes what is the Ka, means we have seen P, we have seen H, we have seen everything, but what is Ka? If you consider what is Ka, somebody asked me what is the Ka? Ka is basically the acid dissociation constant. Okay, acid dissociation means how the acid will be breaking, that is the acid dissociation constant. Okay, basically it measures the strength of the acid. It measures the strength of acid. It is a measure of strength of acid. Measure of strength of acid, that you should know. Okay, okay. Then comes the measurement of pH part. What are the measurements of pH? So pH measurements is basically done by three types of method, that is using pH indicator strips, then using pH meter, then using pH indicator solutions. Okay, so this is simply three types of methods that are used in the pH measurement. Okay, so later on we will discuss the methods in detail, but now we will discuss only the procedure part, how they are being measured in three different ways. Okay, so let us discuss. So the first thing you are seeing here, that measurement of pH is in water and soil samples are crucial. Okay, in various fields such as environmental sciences, agricultural field, and water quality management part. So basically what we need for here, we need pH paper or pH indicator strips like this one. Okay, so material requires basically pH paper or indicator strips, water and soil samples. So what we will do here, for water samples we just dip the pH paper into the water sample, remove the strip and compare the resulting color to the chart provided by the pH paper. These are the chart you can see. These are the charts. Basically these are the charts. So you just compare it with your defined strips that you are getting. Okay, then match the color on the strip to the corresponding pH value on the chart and say it is this pH is this. Okay, this is very simple. Now for soil sample, what we will do, mix a sample, a small amount of soil with distilled water to create a paste. We will create this paste. Then advantages, simple, quick and inexpensive. And what will be the disadvantage, less accurate compared to the other methods in limited range. Correct, this happens. So in this method 1, this is pH strip. These all are pH strip. Okay, then comes your measurement of pH. Measuring the pH of the water and soil is crucial in various fields such as environmental science, agriculture and water quality management. This is method 2. Here we have pH meter like digital, analog, where pH buffer solutions for calibration is used. So what we will do, first we calibrate there are some tablets. Okay, there are some tablets, standard tablets of different pH like pH 4, tablets like pH 7, tablets like pH 10. Okay, So what we will do, water, soil and electrode are used for cleaning the solutions. Then before measuring, calibrate your pH meter using buffer solutions. We will calibrate it buffer solutions of known pH values like pH 4, 7 and 10. Then we follow the manufacturer's instruction for calibration steps. For water sample, submerge the pH meter electrode into the water sample, stir gently if the necessary if necessary to ensure the uniformity. Wait for a stable reading. Then we will wait for a stable reading to get. Then for soil sample, we'll mix one part of the soil with one part of distilled water to create a suspension. Okay, and then allow it to settle for 10 minutes. Submerge the electrode into the suspension without touching any solid particles. Wait for a stable reading. So all these steps we are very easy to remember. We just do it for your measurement of the pH. Then cleaning. After each use, clean the electrode with distilled water and then with an electrode cleaning solution if recommended by the manufacturer. Okay, so this is very simple steps while we will doing pH measurement using pH meter. Okay, then comes titration method. What we will do in titration method, we will use burette, pipette, standardized strong acids and base and solutions like HCl and NOH as an indicated dye. Okay, like phenolphthalein, methyl orange, water or soil samples are also needed. So for example, the procedure here we are that for water sample use them directly. For soil sample, mix 10 gram of soil with 50 ml of distilled water and shake well before the settling. Before the setting we will do that. Then what we will do in titration step, transfer a known volume of what your sample into a flask. Add a few drops of indicator dye. Few drops of indicator dye will be added. Then what will happen in titration process? Slowly add the standardized acid base solution from the burette while stirring until you observe a color change indicating the end point. Then comes record the volume of titrant used at this point. Then calculate the pH based on stoichiometry. Means just we have to see the endpoint. First we will do this. Here is the endpoint. Then here we will getting on the endpoint. There will be a color change during the two endpoints, and we will get this volume. Then we will just measure it and say this much amount of the sample is needed for calculating the pH. That's all. There is not a big theory for this kind of titration method for pH. Okay, so we just use this method for pH calculation also. Okay, so these are all the steps that we have seen for the pH calculation, and this is a very simple 3 to 4 detailing of the pH and how it is calculated. I hope you have understand all the things, and thank you very thank you. Have a nice day. And these are the details where you can contact for regular classes and get good marks in the exam. And please read these presentations and follow up this presentation, and do subscribe our channel for more and more details, and go through these stuffs for your exams that are being held in your universities, as well as the practical knowledge about the pH, which is regarded as the power of hydrogen. Thank you again. Have a nice day.